Okay, next we look at the process of translation elongation. Right, so in the previous video, we have talked about how the translation initiation complex is formed. After that, elongation of the polypeptide chain would begin, and the process is similar in both pro and eukaryotes. Alright, so at the end of initiation, when the complex, the initiation complex is formed, the initiator tRNA, the first tRNA carrying the first amino acid, whether it's methylene or formamethylene for the prokaryote, this amino acid tRNA would be found at the P site. The A site would be empty, and the A site is positioned at the next codon on the mRNA, right? So the next amino acid tRNA would come into the A site with and the next amino acid tRNA, of course, must have the anticodon that's complementary to the next codon. Okay, so not any amino acid tRNA can come in, right? So with this, right, with the matching of the anticodon of the tRNA to the codon, right, and remembering that during preparation, a specific amino acid is added to a specific tRNA with the anticodon, right? So this makes sure that the correct amino acid, as coded by the codon, right, is brought to the ribosome to be added to the polypeptide. Okay, right, so after the coming in of the next amino acid tRNA, a peptide bond would be formed between the amino acid at the P site and the amino acid at the A site, and that is catalyzed by a ribosome called peptidyl-transferase found in the large subunit, where a ribosome is an enzyme that's made of RNA, right? So in this case, it is made of the rRNA found in the large subunit, right? So this enzyme is not a protein. This enzyme is like an RNA molecule, right? And it will catalyze the formation of peptide bond, and once that is done, right, now the dipeptide, right, in this case, is attached to the tRNA in the A site. Right, so the bond between the tRNA in the P site and at first the methylene, right, is broken. All right. Next is the translocation of the ribosome, which requires energy from GTP, right? So the ribosome would move along the mRNA in the 5' to 3' direction with respect to the mRNA. Okay, so now the free tRNA, right, at first in the P site is shifted to the E site, and it will exit the complex. Right, the tRNA carrying the dipeptide at first at the A site is now at the P site, and the A site is empty for the next amino acid tRNA to bind. All right, so the next one will bind. Let's just go through this cycle one more time. And this one will bind, and then you form the peptide bond via the peptidyl-transferase, and now the tripeptide is attached to the tRNA in the A site. Translocation happened again. The free tRNA leaves, and the tRNA carrying the tripeptide is in the P site, A site ready for the next amino acid tRNA. So imagine this process just keep continuing until the stop codon, right? Then you have a elongation of the polypeptide, right? And the elongation is in a particular direction as well, right? The new amino acid is always added to the carboxyl end, the carboxyl terminal, right, of the growing polypeptide. Okay, so in this case, right, you can imagine, right, how the mRNA sequence as a sequence of codons is used to produce the amino acid sequence, right, via the bringing in of the correct amino acid by the correct tRNA, and that is and the ribosome makes sure that the tRNA is correct because of the complementary base pairing between the anticodon and the codon. Right, so the correct tRNA would come in, and the correct tRNA should carry the correct amino acid as controlled or as determined during the preparation step because of the enzyme specificity, right? The specificity of the amino acid tRNA synthetase, which recognize the specific amino acid and the specific tRNA. Right, so all these mixtures helps to ensure that the amino acid sequence is correct. All right. So a bit of extra info, right? How does the translocation occur, right? The translocation occur when the GTP binds, right? It causes a conformation change. And when the GTP is hydrolyzed, that causes another conformation change. And lastly, when the GDP is leaves the complex, it causes one more conformation change. So this series of conformation change, right, and results in the ribosome moving along the mRNA, right, and also resulting in the exit of the free tRNA from the E site, right. So it's a series of conformation change of the ribosome, right, due to the GTP binding and hydrolysis. All right, that's why, as mentioned, translocation requires energy from GTP hydrolysis, right? So lastly, let's look at the termination process when the ribosome A site especially reaches the stop codon. Okay, so instead of a amino acyl tRNA entering, a protein known as a release factor, right, enters, and the release factor carries a water molecule. All right, so what happens is the water molecule would be used to hydrolyze the bond between the polypeptide and the tRNA in the P site. So the polypeptide, now that it is completely formed, would leave and then fold into the correct conformation for the protein, and the entire translation complex would dissociate. And that's the end of translation. And this is similar in both pro and eukaryote. All right, so that's the process of translation as a whole. And now a question to kind of sum it up. Right, so spend some time looking at the question, and you can resume the video. You can pause now and resume once you're done. All right, so let's look at this part by part. All right, part 1, right, label 5', 3'. Right, so remember that the ribosome moves down the mRNA in the 5' to 3' direction, right? So where the ribosome bind to would of course be closer to the 5' end, so that it can move along, right, in the 5' to 3' direction. Right, the start codon AUG would be the first AUG that is after the ribosomal binding site, right? And then, right, to translate this mRNA, right, according to the codon table, right, as covered in genetic code, okay, the AUG would be translated to methylene if it is a eukaryote, right? If it's prokaryote, it's formylmethylene, right? The first one is modified. Okay, after that, the next codon, non-overlapping, right? So the next codon is UAU, which codes for tyrosine, then GGU, which codes for glycine, GGG, which codes for glycine again. We call degeneracy of the genetic code, right? CCA, which codes for proline, and UAA as a stop codon, right? So not really a long polypeptide, a short peptide containing 5 amino acids is synthesized from this hypothetical mRNA. Okay, so this question should help you to consolidate the translation process without thinking about the ribosome itself, right? And of course, what you have learned in genetic code. Okay, so how to read the mRNA during the translation process. Okay, and of course, again, the genetic code, the rules, right, to use to read the mRNA. Yeah, can help.